Hello, everyone, and welcome to today's webinar, Biomarkers in Traumatic Brain Injury and Concussions, Finding Novel Biomarkers. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SomaLogic. To learn more, please visit somalogic.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Jessica Gill, Bloomberg Distinguished Professor at Johns Hopkins School of Nursing. Dr. Gill, welcome. You may now begin your presentation.
Great. Thank you so much. It's really exciting to be here today and to be able to talk about some of our research and to really kind of give an overall perspective about how biomarkers are now and where we're wanting to go. And so, we've really been looking at so many different ways to find novel biomarkers. And so this is just an area where there's so much growth and so much potential. So I'm hoping to get lots of really great questions. So please send them in as it goes along. The first thing that I want to talk about and just present some of the findings, and this is really where we're looking for novel biomarkers. So I'm one of the investigators at NCAA Care Consortium. One of the really distinct values of this is that it includes a high number of universities, as well as one of the best things that we have in biomarkers is to be able to collect a baseline biomarker on an individual, because we know that individual variability really contributes to what we see after concussion. And so just to give an overall idea about the design of this study is that at these universities and as well as service academies, so we have 8 universities and 2 service academies, both the Air Force as well as West Point, we are able to get neuropsychological functioning, balance, as well as a blood biomarker before they have any type of collegiate or training-related experience. Then during this time, if they do have a concussion or suspect to have a concussion, we can get blood on them as well as their age, gender, team match control, and in the military service, also with them, if it's training related or if it's sports related. And so this allows us to look at individual variability as well as to really do the impact overall of timing. One of the other really great things about this study and the design itself is that we have individuals who are engaged in sports and are active, but are not at risk for subconcussive hits, as well as brain injuries such as concussions. And so that allows us to look at the impact of exercise and really minimize that while also looking at the impact of concussion. So it gives us a great kind of lens into identifying biomarkers that are specific to concussion. And in this cohort, we have used a number of different things, and I'm going to present just some of them now. All the findings I'm going to talk about today are based on the demographics that you can see there. But we've now gone on to do the rest of the CARE consortium. So we kind of call this CARE 1.0, where recently we've completed all of the biomarker findings for the second phase, but we're still just in the process of analyzing. So I'm going to show you some of the findings that we have and have published recently. Sorry, a bit of nose itch. So one of the things that we found, we looked at something called 4-plex. And so that's GFAP and UCHL1. Those are the 2 biomarkers that are identified by the FDA as the diagnostic biomarkers of concussion. But just to kind of bring it up now, in this FDA finding, it was really identifying those individuals who had a CT positive finding versus healthy controls. And so the nature of that is really one type of neuroimaging finding versus a healthy control. And so in these studies, what we're doing is we're looking at individuals that have a very subtle injury. So a concussion, which generally would not bring somebody into an emergency room or to seek care. These are things that are witnessed or observed by a trainer and thought to be like, wow, we really should look at it. But it's not something that necessarily, means that you're going to get neuroimaging to have a neurosurgical intervention. And so it's really the more subtle things that we see. And so what we can see overall is we kind of replicate those first findings, finding that GFAP as well as UCHL1 are elevated in the acute period. And this acute period is anywhere between 2 and 24 hours. We also see overall that we get elevations in that of tau, but we find that there's a reduction 24 hours later. So this is something, as well as GFAP, that we've been looking at to really understand the impact of exercise. And I'm going to show you some findings related to that. We also are really interested in biomarkers that are prognostic and that they give us the ability to identify that 10% of individuals who will go on to have lasting symptoms or deficits after a brain injury. And that's really the focus of a lot of our work, because we know that we need to diagnose TBI because, in a lot of settings, we don't have the ability to have neuroimaging or in-depth phenotyping. And so having a biomarker that can say, this individual had a brain injury is really important. And especially in individuals who are in the military or in sports fields, because those are the individuals we want to follow. But really, the individuals that we want to follow the most is not those that just had the injury itself, but those that are more likely to go on to have lasting symptoms and deficits. And so we've done a lot of work on return to play as well as return to duty. And so these are some of our findings here. The first initial finding is that tau is elevated, especially acutely. So we see reductions overall, and I'll give a little bit of insight into that, because what we found is it's really related to a lot of physical exercise and other factors, but really understanding the impact of that. So we did that in the first study, which was in 2019. We were able to expand this into the NCAA Care Consortium and find that we were able to replicate tau as being a predictor of return to play, such that the higher levels indicated that those individuals were more likely to have a prolonged recovery, and that they'd have more than 14 days in which they had to be resting and not be able to come back to play to be fully recovered. One of the things about return to play that's really important is that if an individual does not have a full recovery before they come back, they're about 7 times more likely to have another concussion. And so that's based on a lot of different factors, such as reactivity time and things that we're not really great at measuring right now. So having a biomarker that says, this individual is more likely to be recovered and can go back to play and be safe is really important. Because what we know from our studies is that if an individual has a concussion and goes back to play and then has a subsequent concussion, those are the individuals that are most at risk overall. And so that's something that we really want to use as a way to make things safer. And it's not just that we want to eliminate any sports that include concussion, because that's a lot of the identity. I played collegiate soccer, and that's something that was really, really important to me. And so we don't want to limit that, but we want to make it safer, especially when we think about military. So we have individuals that need to have training related to many things like firing a weapon, being able to jump from a plane. And so that training often includes a high risk of having a concussion. And so what we want to do is, if you do have a concussion, we want to treat that the best way we can and to minimize secondary injuries. And so what we're seeing here is that tau, as well as GFAP, which was our secondary finding, and more of a trend, which we're now following up on, was really important to look at related to recovery. And so that's some of the stuff that we're looking at to see what predicts recovery and how we can optimize the care that we provide. We've also looked at phosphorylated tau, and specifically what we've looked at is 181 as a specific isoform. And the reason that we look at this is tau overall is kind of an umbrella of a lot of different isoforms that we want to understand better, because it's not just tau elevations that are related to this kind of recovery process, but it's the phosphorylation of tau that we believe is most important. And once tau becomes hyperphosphorylized, it becomes something what we call kind of like a sticky protein that is hard for the brain to clear. And once there's aggregation of that p-tau within the brain, it predicts long-term outcomes in those individuals, such that they may be more likely to have something like chronic traumatic encephalopathy or some type of other cognitive decline. And so a lot of our studies, both at NCAA, especially as we're going now into phase 2 and now phase 3, where we're able to follow these individuals over time. And right now what we're doing is we're following individuals after they graduate and as they go into kind of their adult life to see how these concussions actually impact overall recovery, but also their ability to have a job, relationships, and things like that, to really understand the long-term overall impact of these concussions on these individuals. And so this is really kind of a hallmark study to understand how these injuries, as well as repeated injuries, can relate to overall recovery, but also kind of a trajectory in life. And so that's where we're engaged now. So hopefully in the next couple of years, we'll have some more findings related to this. But overall, what we see is phosphorylated tau occurs, especially within an acute period, 0 to 6 hours. And from preclinical studies, we know that there's a massive translation of this, such that phosphorylated tau occurs within a couple minutes to hours. And so it's really important to know when it's happening, but then also when we can also detect it in blood and do that sensitively. And so, we've had a lot of success looking at these candidate biomarkers. But again, what we know about GFAP and UCHL1 is they're based on that CT positive finding, which is not often observed in individuals who have more subtle injuries like a concussion. And to give a little bit of a perspective, when we looked at studies within the CARE consortium, we find that concussion very rarely relates to a CT positive finding, less than 1%. And so we want to look for biomarkers that are more based on the pathology that we're seeing, both in the subtle injury as just the different nuances of the type of injury. And so what we've done is try to find biomarkers that more approximate this. And the end goal of this is really, if we know what biomarkers are mapping onto this, it gives us an ability to identify them and then use them to monitor those individuals who've had a concussion. But even more important, it gives us really fundamental insights into the pathology that relates to it. So understanding what's going on in these individuals is essential, as that we can then look and see what interventions we can then modify or use to intervene. Because what we see is once individuals have the development of symptoms, especially in a chronic nature after 3 months, is that there's neurological findings related to MRIs that indicate that there's changes. And once those changes happen, it's very difficult to reverse. And so if we can identify those individuals early who are more at risk for this and then to intervene early, we have a really great opportunity to modify those risk factors so that those individuals don't go on to have those long-term symptoms. But our current biomarkers, of course, are limited. They're based on that CT positive finding. And so for our goal, we're thinking, how can we expand this? How can we identify new biomarkers that give us that identification that they've had a concussion itself, as well as an identification of those individuals who are more likely to have those lasting symptoms and deficits? And so this is why we've used SomaLogic to do that. And so instead of just looking at a candidate approach, as we would in ELISA or something, an antibody, we can look at, up to 7,000 proteins at the same time. And that allows us to look at different pathways, not just a protein, but things that are up and down regulated. And, the down-regulated proteins are really difficult to understand because usually what we're looking for are elevations in proteins that give us a signal. But this can give us kind of a map of what's going on. So we can identify proteins that are both up and down regulated, but also hub-related proteins that give us identification of pathways. And so this opens up a whole new avenue. So we're not just sitting around a table and guessing that we should look at this protein or that protein based on some preclinical model or hypothesis, but that we can actually use the data to kind of mold us and to guide us to look at what's most important. And so this has been a lot of the function of my lab, is to use these non-hypothesis approaches to identify new biomarkers and to really go after them and understand their relation to the brain injury and the overall recovery process. And so what we do is we look at canonical pathways. So this is a way to see kind of where the dysregulation is occurring. And then it's not just one protein, but it's maybe a set of proteins that we want to look at. And the value of this is we can identify a dysregulated protein and a pathway. And then we can identify interventions that would then go in to address that specific pathway. And so we use this in a way to be more prognostic, in that we want to understand what's going on in those individuals, again, both diagnostically as well as prognostically. And so one of our findings, so we did it also within the CARE consortium. And so, the whole idea is that GFAP and UCHL1, as well as neurofilament light chain, they give us a lot of ability to look at it. But when we look at the findings, we only get area under the curve of diagnostic accuracy of about 0.8 up to 0.2, 0.3. So what we see when we actually use the SomaScan technology is that we could identify new proteins, again, that probably map onto that pathological or the physiological changes that are related to the concussion. And so here we have a finding. So these are proteins that we wouldn't even look at. So the first one is erythrocyte membrane protein band 41. I would have never looked at this. So we call it EPB41. So we see an upregulation of that protein after concussion. And so just to give an idea, so these are individuals within 6 hours and 24 hours after a concussion. And we look to see which proteins specifically on the array. And when we use the array, we were at 2,500 proteins. It's now been expanded to much more, but that's the platform we used about, 18 months ago. And so what we found is it gave us a really, really great estimation by combining them. But EPV41 overall was great, just having that one. But adding alpha-synuclein, and so this is a protein, again, that's downregulated, which we normally don't know how to deal with this as much when we put it into our models. But we see that there's a downregulation overall in alpha-synuclein. When we combine EPB41 with alpha-synuclein, we get a prediction that's almost 100% of diagnostic ability in concussion. And again, just to remind you that these are individuals that have a concussion during a sport. And so they're not necessarily reporting to emergency rooms and saying, I've had a concussion, but these are things that are observed that the individual might not even know that they've had. And so this is really the most subtle form of concussion. And so, going back to the FDA finding, the combination of UCHL1 and GFAP did not get this performance. And those were individuals that had much more severe types of injuries. So it really gives us an ability to look at specific proteins that are up and down regulated, as well as the combination of those, as well as networks. And just to kind of give a little bit, so this is some of our findings when we've done these. So seeing the CT positive finding, we do find that those same proteins are elevated. But in this study, we added an MRI subgroup. And so these are individuals that had both a CT and MRI. So CT is the standard of care, but it only really has that one diagnostic profile that indicates that you have to have neurosurgery to remove that kind of blood clot or aggregation of blood. But having an MRI finding opens a window of pathology. So it can be anything from microbleeds to breakdown of the blood-brain barrier, things that really require a more sophisticated way to look at it using this advanced neuroimaging. And so it becomes, again, kind of like tau, an umbrella of different pathologies. And so I'll present a little bit more on that of one of the sub ones that we've been looking at more intensely, because it's really important to understand the clearance of proteins. And so overall, we replicate that finding, but again, we extend it to this group that has no CT positive finding, but it does have an MRI positive finding. And the significance of this is that those individuals that have this finding on MRI go on to have less ability to recover. But these are also individuals that leave a standard emergency room and feel like they haven't had a brain injury because they had a CT positive negative. But these are the individuals that we actually do want to follow, because we know that their recovery is compromised and that we want to monitor them and provide interventions. And so it kind of extends what we're looking at of just not this one surgical intervention, but all of these different things that we can do for these individuals that go on to have long-term symptoms. And when we look at those individuals, again, we have about like a 15 to 18% rate of individuals who have this mild brain injury, which is thought to be as a Glasgow Coma Scale between 13 and 15. So these are individuals who feel like maybe they fell, maybe they didn't, somebody observed them. There might be substances related to it. So it's a really difficult group for diagnosis when we see them in emergency rooms. And so this allows us to say, okay, you have had a brain injury and you're in the class of individuals that we want to follow. And it kind of makes it closer where we can use precision-based medicine to understand it and to really improve the care that we provide. Because if they are in that group of individuals, that subgroup of that 15% or so, they can have long-term outcomes that relate to compromises in employment, relationships, and overall health-related quality of life. And so if we can identify them and intervene early, then we have an option or an opportunity to really improve their life and mitigate the impacts of traumatic brain injury. And so one of the subtypes that we've looked at is traumatic meningeal injury. And the reason that we see that this is important, first, it's very difficult to do neuroimaging in these individuals, in that it doesn't just require an MRI, which is not available. MRIs are only available in less than 3% of emergency rooms. So it's not a standard care of intervention. And usually when an MRI is underdone, it's people coming in who say, I've had a TBI, I was diagnosed with a TBI, and now it's been 1 month, 2 months, 3 months, and I'm having ongoing chronic symptoms, things like headaches, sleep disturbance, mood disturbance. But again, that's when we've already seen these neural pathways change, and these changes we don't know how necessarily to reverse. And so we're really kind of in a catch-up game when we see them. And so that's most of what we know related to TBI and MRIs is this more subacute finding. But within this study, we have the ability to look at traumatic meningeal enhancement, and that's done through the contrast agent of gadolinium that is given. And so in this study, what we see is the gadolinium gives a proxy of the function of the blood-brain barrier. And what's really important about that is we want to have a blood-brain barrier that's intact in some ways, so that if it's compromised, what we see is that there's infiltration of a lot of immune cells and we get overactivation of the brain. And we don't want to have this chronic inflammatory process. And so what we know about inflammation is that it's very essential for recovery. So in preclinical models, if you knock out inflammation altogether, you won't have recovery. But if you knock out the blood-brain barrier, you can have really detrimental effects completely. And so it's really the balance of inflammatory activities that then initiate neuropeptides that facilitate both neuroregeneration as well as clearance of these proteins. So we want proteins like tau as well as amyloid beta to be able to get out of the brain, but we don't want so many immune cells coming in that it disrupts the recovery process. And so it's really a balance of severity as well as time. And so if it's very severe and the blood-brain barrier is not intact, we can see infiltration of all these different immune cells and overactivation of the immune system. If it ends up being prolonged, then we can see overall chronic inflammation, which has specific deficits that I'll show in a little bit. And so it's really trying to understand this and not just the impact of inflammation, but also this is really important for clearance. And so after a brain injury, we want to see degradation of proteins that relate to recovery. But if the blood-brain barrier is intact and not letting those proteins go out, you can have aggregation and you can have the onset of things like tangles and plaques, which we know is a hallmark of CTE. But we also want to have clearance, but we don't want to have infiltration. So it's really a balance of these activities that's most important. So this has been a big function of our lab, is understanding how these proteins got cleared, both by the meninges, as well as perivascular space, which is a proxy for lymphatic function. And so that's a series of studies I'd be happy to talk about, but we don't have findings now because it's a lot of the work that we're doing. So overall, we can see this finding. So this is here, and you can see the enhancement of the meninges, specifically after brain injury. And in this individual that we're showing, this brain injury and TME was observed within 36 hours. And then when we saw the individual again at 5 days. And so the thing about TME also is that you really only see it acutely or subacutely within 24 hours to maybe a week after. And so it's kind of a pathological finding that we're starting to understand the mechanisms that relate to it, but because it's a very difficult procedure to have done and to get this data, it's limited. And so what we're trying to do is find biomarkers that approximate TME, so we can use those in other studies, and we can identify mechanisms that relate to this, so that we can have a biomarker, because having these imaging both is difficult if there's not an MRI there, and that's not going to become standard of care anytime soon, as well as to understand the nature of it, as well as how it relates to clearance and the lasting nature of it. And so when we look at some of these findings, we see overall that we have some indication that those individuals who have that meningeal injury have elevation of those same biomarkers. And so that gives us indication, but again, probably can we get better at this? Probably, because the biomarkers that we looked at were in that fourplex that were more related to TME, more related to the CT positive finding. And so by using more novel-based approaches, we can actually identify TME-specific biomarkers. And so this is a big project within our lab, as well as within the intramural program at NIH and NIDS, and something that we're looking at, because what we see overall if individuals have this TME finding acutely, but more importantly, if they have it and they continue to have it at 5 to 7 days later after the injury, those are the individuals that go on to have long-lasting cognitive declines. And so we know that it's really a prognostic indicator, but it's not going to be easy to measure using this MRI method. And so can we find a biomarker that approximates in a better way and then use it within clinical care? So that's really the focus of a good amount of our studies within our lab. The other focus within our lab is how can we use blood to get more specific? And so the area we've looked at is using exosomes. And so exosomes are nanovesicles that are secreted from every cell in the body. When it's secreted, there's an antibody that's put on the outside of it that allows us to identify which cell type that it came from. And so we can use that antibody to then say, this is brain specific or microglia specific or astrocyte specific. And so as we get more and more evolved in this, we can actually say what's centrally related to the brain, because that's really what we're trying to do in these studies, especially mild TBI as well as concussion. These are not patients where we can get a CSF level or we can do anything really in depth. And so we want a method that allows us to get more close to the brain without being intrusive in a clinical nature. And so this allows us to do so. And really the important part about exosomes is when that exosome is secreted from a cell, it then goes to bind to another cell. So you have a brain-to-brain connection, specifically for what we're looking at. And the contents of the exosome are really important because of a brain. So if an exosome has more inflammatory dominated, we can see inflammation spreading from one brain cell to the other. This is particularly important when we look at microglia. And so it gives us an ability to kind of look into the window of what's centrally related to understand those mechanisms without getting very invasive within a patient. The other really important thing within brain injury is it's important for clearance of these proteins. So after brain injury, we see elevations in GFAP, tau, all of these different proteins, as well as amyloid. If they stay within the brain, they can have translations of those proteins that make it more kind of sticky. So they're going to stay there. And when they stay there, they can compromise the functioning of that neuron, such that that neuron is marked for degradation or clearance or death overall. And so what we see in TBI, when we really see deficits in a chronic nature, is we have black holes in the brain where there's areas where neurons are no longer connecting and that area of the brain is no longer functioning. And so what we want to be able to do is say that this is where that it's happening. And we can actually develop mechanism-based interventions that allow us to go in and be specific in the way that we're intervening. So right now with TBI and concussions, there's no medication that facilitates recovery overall. And so what we know is that the pathology is very diverse. So everybody who has a brain injury has a different type of brain injury and different pathology. And so using exosomes, we can actually get in there and see what's going on and understand it in a better way. I'm going to show you a couple examples of that. And so this is one of our findings. So what we did is we did breachers. So breachers are individuals within the military who have thousands often of experiences of blast exposure. So blast exposure, what we're coming to learn, this is a lot recently, is that blast exposure is not great for the brain. So it's somewhat similar to what we think about subconcussive hits or subconcussive exposures. And so these are individuals who are training other individuals to fire something or to dispose of a bomb. And so there are people going in and breaching, we call it like breaching a barrier. So they're coming in and they're saying there's a bomb, boom, I'm going to come in. And so they're exposed to high numbers over their career. And so part of the military and the Department of Defense was to come in and say, we've got a cohort of individuals who are really compromised and concerned about their health, because these are individuals that are highly functioning, selected because of their intellectual abilities, but then over time they're compromised, or at least they're reporting compromise. And so we did a series of studies at NIH, as well as continuing to look at these individuals and understand the nature of brain function and really what's going on related to this. And so this is one of the findings that we had. So overall, when we look at the blood, we see that there's really not that much difference when we look at cytokines and things like that. And so just give kind of a background. Anytime we have a study in individuals who've had TBIs or depression or PTSD or have combat exposure, we see an elevation in cytokines. And those elevations relate overall to psychological as well as neurological compromise. And so when we did this study, we thought, well, this doesn't seem like this doesn't make any sense because their elevations are not there. And so what we did is say, okay, the signal is not there in the blood. Let's go more specific and use these brain-derived brain-derived exosomes to look at the contents and specifically of cytokines. And then we also did some of the neuronal biomarkers. And so what we found is that overall, the inflammatory signal is not there in the blood, just the general blood. When we look at cytokines, we actually see them very elevated in brain-derived exosomes, as well as elevations in tau and other biomarkers. And so it kind of gives us a window to say, this is something that we should be worried about because overall neuronal inflammation is one of the biggest predictors of Alzheimer's disease and other neurological compromises. And so we need to be able to really understand that. And so that motivated us to undertake a study to look at specifically the nature of brain inflammation measured by a PET ligand. And so that's not the one, but so when we did that, we were able to really isolate it. So this led us to a different study finding. And I'm just going to give one also idea about what we did. So this is in a different cohort, but we were able to look at specifically lasting neurological symptoms and microRNA within exosomes that were brain specific. And so we see a specific pattern related to this dysregulation. So what you see are dysregulation networks related to microRNA. And so we see that this is a way to really look at these chronic symptoms and understand the nature of these. This is really clinically important because when we see veterans, we have a number of interventions we can do, but they're not necessarily pathologically based. And so how can we find these individuals and really understand the pathology that underlies the symptoms that we see? There's also a lot of comorbidity in these individuals. So how can it kind of indicate networks that are not just related to brain injury, but also PTSD and depression, as well as sleep disturbance? And so that's a lot of the work that we do. It's not going to be, this is the end all medication for treating TBI in a chronic nature, but this is what we see in clinical populations. And this is how we want to intervene the best way. And so then it also led us to a study where we can look at central inflammation. So this is really exciting to me. So we can use PET ligands. So PET ligands allow us to look at a specific pathway. Here we use TSPO, and this is James Stone's work at UVA. And so we were able to use this and to look at brain-related inflammation, and then we can see where in the brain it is. Then we can do our different protocols to look at exosomes and make it specific to see if inflammation is there within those exosomes. So again, it makes us more and more close to the brain, but this is the first window in where we can get to specific parts of the brain that allow us to identify proteins that are dysregulated as well as microRNAs and say, if we really can change this, we can change brain function in these individuals that have chronic symptoms. And just give kind of an idea about these types of patients. And so the ones coming in and saying, I'm just compromised in my sleep. I can't work. I'm having problems with my relationships. And so there are a lot of vague type symptoms, but if we can get down to a pathology that we can understand and intervene, it gives us a whole new window to really make an improvement in their quality of life. And so kind of putting all this together, our current limits when we're looking at proteomics is, first, and I'll show you a slide or two on this. The time course of proteins is important. So what I always say, it's not just the right biomarker, but it's the right biomarker at the right time. And so there's very few studies that allow us to collect multiple biomarkers on the same person over time to really understand the temporal relationship of this and some of the other variables that I'll talk about. But this is really what we need because, when we see people in combat after concussion, as well as in the emergency room, they're coming at highly variable times. And so it's not going to be one biomarker that's going to say, yeah, you did or you didn't have a TBI or concussion, but it's really a combination of the biomarkers and also the understanding of time and how those are changing. We're also facing the technology of how we can measure these very low proteins, especially in clinical settings. So, we're looking at things like, grains of sand in an Olympic-sized swimming pool. How can we use this sophisticated technology to understand these small quantities that are leaking out from the brain. And that's always going to be one of our complications of how we can really get the best signal that we can, both in blood as well as other biological fluids. Another thing is mapping objective fluid biomarkers on imaging biomarkers. Again, what we want to do is be able to see an imaging biomarker and say, okay, now we've got a fluid biomarker to approximate this. But because of all of the issues with individual variability as well as timing, this is not always accurate. So the pathology that's occurring within the brain has a delayed effect of what we see in the blood. And that relates a lot to the clearance, which we're trying to understand. So we can have a pathology within the brain, but we're not going to see that protein unless we have a blood-brain barrier that allows for that protein to come across it. And so, having something like when we look at creatinine for urine about the clearance factor would be extremely essential to understand that it's not that we're not seeing it, it's just that it's not there yet. So how can we understand the timing and also, the confluence of this barrier that we're seeing both in blood-brain barrier as well as lymphatic function. Also understanding of how proteins are related to the origin. Many proteins I think that we identify are great if we want a diagnostic biomarker because they allow us to understand that something's going on. We need to focus on that. But they're not necessarily related to pathology because there's so many translations of that protein that when we see something, it doesn't mean that if we intervene based on that protein that we're going to have really an impact. And so our lab specifically has really been related to understanding multiple time points and then also, canonical pathways and hub proteins to really understand it's not just this one protein that's up or down regulated, but it's a protein that seems to be influencing a number of pathways that we think are related to this. And that's not always easy. And what we want to do is within the same individual, see that same protein or that network changing over time and relating to something that we can see in neuroimaging. And if not that, at least within patient reporting of symptoms or objective things like neuropsychological functioning. Also, I'm going to show a little bit of data about I think one of the things, and this is kind of a paradigm shift that it's happening within TBI, is that we used to think about TBI as kind of like a broken bone. And so we can see it, we can measure it, and then we can cure it. But within TBI, it's very different because many of these injuries that we think about TBI or concussion are related to traumatic events like intimate partner violence, child abuse, and other things like community violence. And so we really need to reconceptualize that, to think about this as not just a specific injury to an organ or a bone, but also to a whole stressful response. Because this response, if somebody comes into an emergency room and has this, it also means that maybe they're going to lose insurance because of employment. And so there's a lot of things that can make recovery difficult. And it's not just neuroanalytical or biomarker based, but it's really integration of the psychosocial factors that we know are so important. And so it's really thinking about, it's not just the injury mechanism, the injury that we can see in the biomarker, we can see reflected in the blood, but it's overall the environment that the individual is living in, including social and environmental factors that really relate to this individual variability that we see in recovery from brain injuries. And so to show a little bit about this, so this is just looking at the time course in individuals who had a mild brain injury. And so you can see these biomarkers vary greatly over time. And so this is one of the few studies that looks at more long-term recovery, looking at these, fourplex proteins, GFAP, UCHL1, NFL, and tau. And we can see that, again, it's not just the biomarker, but it's the biomarker combined with time that's the most important factor and really predicting these overall recovery trajectories. One of the other things that we've been able to look at more closely, especially in our concussion group, is the impact of exercise. So it used to be that when an individual had a concussion, they would just go into a dark room, no video games, no sound, no anything. We found that overall clinically that wasn't therapeutic. And so people go back to exercise and things like that. And so when we're looking at biomarkers, there definitely is an impact of exercise just itself. And so in this study, They were individuals who were collegiate athletes, but they were not engaged in their collegiate sport. So it was off season, and they did an aerobic exercise like running with a trainer, push-ups, sit-ups, things like that. And so we can see differences based on these biomarkers that we think are really brain specific, but actually are not necessarily, because they do have the impact of exercise itself. And so we see, overall some increases as well as decreases within these proteins. And so, coming back within the NCAA Care Consortium, we are able to look at the impact of exercise and also, take it out of the impact of subconcussive hits. And so that's one of those things that we just really need to look at, because we know from a lot of clinical studies that exercise itself is protective and facilitates recovery. And so we want to be able to use that. But if we're using exercise as an intervention, then we also have to think about it as changing these biomarkers that we think about as monitoring biomarkers or prognostic at least. And so it's just really, again, looking at individuals over time and understanding the impact of these overall, activity-related individual variability things that we're really understanding impact recovery over time. I'll just give one more slide on individual variability. It's really, a lot of our studies have looked at gender or sex, and it's kind of an avenue into looking at more sophisticated ways to look at individual variability. But we do find that overall, there are a lot of impacts of this, both in the trajectory of tau, which relates to recovery, as well as these other biomarkers. And so going in and seeing, is it gender itself, which has a whole lot of characteristics which are much more specific than sex, or are there things that make some individuals more likely to not recover? So how can we kind of take it out of this box of yes versus no whenever we go in somewhere for a medical appointment or file our taxes or things like that, to understand the nature of it and what characteristics matter most. And so we see that there's a signal there for sure, but what's the nature of that signal, and how can we understand the components of that signal that matter most? And so that's a lot of the work that we're doing within my lab. And so this is just a conceptual model of kind of how we think about looking at this. And so it's no longer just that somebody has an injury and then they have an outcome, but there's a lot of components that are pre-existing when an individual comes into an emergency room or is on a sports field or has an injury when they're deployed. And so all of these pre-existing factors can dysregulate many of the functions that we think within the brain as well as systemically that relate to recovery, including neurological changes, which can make individuals more likely to have neurodegenerative processes, as well as the regulation of immune function. And so we think about this kind of on a lifetime trajectory, that it's not just one injury, but many individuals that have one TBI are much more likely to have another based on their factors. And so we want to really understand the impact of this lasting symptoms and how we can intervene to best really modulate those to promote recovery. Because overall, in any type of study we do, we see, TBIs are related to health morbidity, health mortality, employment, just about any type of outcome you can think about being negative. TBIs are associated with that. But it's not just TBI itself, but it's the way that the individual interacts with it. And so how can we understand those factors in a more comprehensive way to design interventions that are going to be more meaningful for those individuals and really make an impact? And so that's really the function of our lab. And with that, I'll take questions and thoughts, and I'd be really happy to get anything that would be important to add to the work that we're doing. And I'll just do also a plug that everything we do has trainees related to it. So if there's anybody who's looking for training opportunities, please let me know. We're always happy to do it remotely as well as in person. And thank you so much for your time and attention.
Thank you so much, Dr. Gill, for your informative presentation. Joining us today for the live Q&A portion of this broadcast is Dr. Dion Denmark, Senior Director of Translational Medicine and Clinical Development at SomaLogic. We will now start the live Q&A portion. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Now let's get started. Our first question here is, have you compared the quantification results obtained by ELISA and by SomaScan? Are they correlated?
Yes, so we have done that specifically. So EPP41 has been something that's been elusive to us because unfortunately the antibodies are not well established for that one. So that was the one that was really predictive of diagnosis. So that has been limited. We're still doing a lot of work related to that, and we're trying to find different ways to do that. But what we have been able to do with alpha-synuclein is to do the same thing and to use the Quanterix platform to validate our findings. I didn't show them here because we're still running it and analyzing it. But we do see that the elevations related to the signal we see within SomaScan relate to this other, antibody-based approach. So that makes us highly confident in what we're doing, and that. And I didn't present a lot of this, but alpha-synuclein it provides a lot of diagnostic accuracy, but it's much more important prognostic. And so we're doing a lot of studies using that, looking within the CARE Consortium, as well as other studies that we have ongoing. So overall, we found that it does align. Our limitation has been if there's not an established ELISA, we have to do a homebrew on one of our methods. And so that's just been one of the things that we've seen be difficult if there's not an antibody that we can use to develop those homebrews.
Thank you for that. Our next question is, how many of your patients have negative side effects from the gadolinium, and how many patients does the use of gadolinium exacerbate the TBI symptoms?
So we have looked at this a little bit because I know there's some controversy with gadolinium. Again, that's why we need a biomarker that approximates that meningeal injury. We have not seen any significant impairment. So within the study, most of the individuals, so we get them acutely, we look at them, and then we follow them at 5 to 7 days. And then there's a cohort that we also follow at NIH for up to a year. We have not seen any negative side effects that are attributed to gadolinium that we can see. But again, that's a little bit flawed because anytime you have a prospective study in people with mild injuries, you more likely see those individuals who are symptomatic coming back. Those individuals who are healthy and just kind of want to go on with their life don't come back as often. But from what we can see, we don't really see too much of an impact. It's not something that we're substantially worried about. But again, that's the reason we want to have a biomarker to approximate it.
Thank you. Another question here, how do these findings compare to any of the known or previously seen biology for concussion or TBI?
So our findings with SomaLogic in that really mild cohort, we got an area under the curve, meaning that we were able to approximate it with 100% accuracy using those two biomarkers. That compares with what we did with GFAP and UCHL1 primarily. We did use some mixed modeling, including tau as well as NFL, but it really was GFAP and UCHL1 that drove that. But the biggest accuracy we could get was up to 0.83 in our area under the curve. So it definitely improved it overall.
Great. Next question here. What were the most exciting and or surprising findings or insights from applying an unbiased proteomic approach here versus pursuing hypothesis-driven biomarkers?
So I'll just say I've been very privileged. So my experience was at NIH in the intramural program. And so I was not focused on grant writing where you have to have a specific pathology or biomarker set that you're looking at. And so my approach is probably different than a lot of scientists who address this because I've not been forced to say it's these 5 biomarkers and this is the budget I need to look at them. Anytime we do anything in our lab, we run 4-plex data on every sample, and that does give us some information. But for example, one of the studies we did with blast, so we did individuals who had a blast exposure that was something you would call a moderate blast exposure. We actually saw reductions in the same proteins that we see increases in. So that means that there's a different pathology going on. And so by using this type of technology, we can go and say like, okay, we're looking at the wrong things. And so how can we get this, to really understand the pathology of that? And so by using this, we were able to identify different protein pathways and then do some preclinical models that were much more informative to understand. Because the issue, especially with TBI in military personnel and veterans, is that blast exposure almost always co-occurs with a brain injury. And so there's a blast that results in a blunt force injury because the individual is thrust into, some type of contact with something else, and that results in a brain injury. And so you're looking at two different injury mechanisms, which is much more difficult to understand because the pathologies are different and the mechanisms that we understand don't necessarily overlap. And so it's stuff like this that allows us to go in and say, like, we don't really know because, blast injury has been pretty new in the last decade. And so there's not established biomarkers related to that. And so we can go in and say, like, we can look at 7,000 proteins and understand it much better than sitting around a table and saying, like, let's test these 5 proteins and these. And then by the time we do that, then the sample's depleted. And so it really gives us an opportunity to go in and say, it's okay if we don't know what's going on, but we can use this to do it better and really inform the pathways and the pathology that we're looking at. So it opens up a lot of windows that we don't normally have. And we do the same thing as when we look at genomics. So we don't look at, 5 different genes, but we're looking across the genome, both in DNA and RNA, as well as microRNA. So anytime I have an opportunity to look at more candidates than people think are reasonable, I do it because that just leads science in a different way. And I think our lab is data-driven, not hypothesis-driven. And so I always say consistently, I'm wrong. Every time I say something's going to happen, I'm consistently wrong because the proteins we look at don't necessarily correlate to the outcomes we're looking at. And so if we don't get that target, we have the ability to go in. And so by going to Hopkins, the reason I did is that I have a foundation that I can use. And so I'm not completely grant driven. And so I can use these really great technologies to say, let's build something new. So you can find a protein and then develop a panel to measure it and then validate it. And that it opens up so many more opportunities than what people do. And I think science is stagnated a lot by the hypothesis-driven approach because you can look at as much literature and preclinical stuff, but all the models are different and it just doesn't lead to new avenues. And so I've been really excited to be able to use this type of thing to say, like, okay, we didn't think about EPB41 being anything related. We never would, but there it is. And it's really accurate. And it's something that we can use moving forward. So yeah, it's a long-winded answer to the question, but it just means that anytime I can use data to guide what we're doing as opposed to going back to literature, we're going to do it because it just allows us to go in directions that we wouldn't have thought of and think like, okay, this just gives us a new window to think about options of how we can intervene. So, yeah.
Thank you. We have some great questions coming in. Someone is asking, have you done replicates and is there some variability between them?
So I would say between SomaLogic, I believe we have. I'm not the best person, so I know a little bit about a lot of things, but not a lot about everything. And so I know that we have done some replicates, especially when we're testing out the technology. So I'll say, when we started using SomaLogic, they were at NIH and they were literally like a floor above us. And so we're like, this is amazing because it allows us to look at clinical translational proteins and really transform what we're doing. So my, the person who was working my lab as kind of like the lab scientist did a lot of work on this before we went into this. And so the quality controls and all of that was amazing. And so I haven't done it recently, but I felt very confident in going into the technology.
Great. Next question here. What are the challenges you have in tracking patient medical or social history over time to provide context for interpreting biomarker results over time?
Well, I think that could be a whole book. So patients are only as reliable as the information they want to provide, right? So I always say, like, anybody who we think is a healthy control, talk to them long enough and they're not. And so everybody's different and different at every time point. And so a lot of our longitudinal studies are tracking that. So I talked a little bit about NCAA. So we're tracking those individuals as they go into kind of their adult lifespan and, the concussion impact. We also have a study in veterans called Limbic, and we're looking overall about the impact specifically of multiple TBIs as well as post-traumatic stress disorder, and how that relates to compromises in health and well-being. But my thought is always that, everybody's resilient. It just depends on when you see them and when you have an opportunity to intervene. But those are going to be things that I think are the challenges for our lab in the next 10 years. How do we measure this the best? One of the things I didn't talk about today is we've developed sweat patches that are highly related to the biomarkers that we see in blood, but allow us to collect them in a way where the person's in their home environment, and then they can actually mail it back to our lab. And then we put it on our same platforms, including Neurology. And so that gives us, again, another window to really understand, how somebody feels like at home, if they're caregiving, if they've had a brain injury, if they have depression. What are the biomarkers that relate to compromise as well as health and well-being, and how can we use that to really inform monitoring, when to bring them in and when to intervene? But that's it's complex, and the more we study about epigenetics too, it's not just, like when my kids were born, I'm like, it's a healthy soul, anything that happened to me before is not inherited. But now the more we're learning, it's not that way. And so there's transgenerational, information about violence and exposure and nutrition. And so my idea is always comes back to biomarkers. Can we use a biomarker to approximate, a resiliency factor versus a vulnerability factor? Because we can't ask all the questions that we need to about somebody, especially in an acute trauma situation. It's not appropriate to talk about, additional traumas that they've had before, child abuse, TBIs, things like that. But if we can get a biomarker that says, this person has vulnerability, we don't need to know the nature of the vulnerability, just that we need to intervene. And so this is, an area that we're very, very focused on. And happy to report, hopefully in 10 years, we'll make progress. I think it's going to take that long to really understand all these complex factors. But I think that's where science is and where we'll make the biggest impact for sure.
Thank you, Dr. Gill. We have time for a few more questions. Another question here is, how did proteomics in particular advance on previous approaches for clinical assessment and understanding psychological consequences of concussion?
So it's huge. So proteomics, we do a lot of different studies in our lab. So our lab is not just proteomic focused, but proteomics is what you're going to use at the bedside. It's what we can measure, and it's what will really change clinical practice. And so, for example, if we get somebody coming in with a TBI, so some TBIs have, a psychosocial focus with them. So it can be assault related, traumatic events, things like that. And so a lot of our work is focused not just on neurological compromise, but psychological compromise. And one of the things we see consistently across our studies, if somebody develops post-traumatic stress, especially if that also is comorbid with depression, that's where we see the biggest impact on their health and well-being. And so that's likely going to be different pathways that relate to that, as opposed to, say, headache or light sensitivity. And so it's those 4 clusters of post-concussive syndrome that we're trying to understand. And it's really through bigger studies. And one of the things, like, starting in this, there were small studies, and now we're at the point where we have consortiums. And so we can look at things that are subtle injuries that relate to these long-term consequences and be much more specific in it. And so we're looking at how these different biomarkers map onto different outcomes. So what's a predictor of PTSD versus a headache? What's a predictor of depression or sleep disturbance? How do they overlap? And then more importantly, how can we intervene? So that way, those individuals most at risk, we can mitigate those risk factors. So I don't have an answer specifically, but it's definitely an area where we're focused on. And hopefully through our consortiums where we have these thousands of people, we can do that work. It's just been recently available to us to be able to do that. But I think that is coming on. And I think, it just really lends it over to personalized based medicine is where we need to be with TBIs and concussion, because everybody has different risk factors based on previous history, as well as the injury mechanism, as well as the pathology specifically of the damage to the brain. And so how can we put that together and say, let's do this? And we think that this is the most optimal, because we really only have that small window of being able to intervene where we're going to have the opportunity to reduce the impact of brain injury and changes overall that really shape the onset of those symptoms, as well as it developing in a more chronic nature.
Thank you so much. And thank you again, Dr. Gill, for your time today and your important research. We'd also like to thank LabRoots and our sponsor, Somalogic, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions we did not have time for today and those submitting during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.